So today we are going to look at the concept of glycogenolysis. So we are starting with the definition and the processes in glycogenolysis. What is glycogenolysis? Is a breaking down of glycogen into glucose, and it is a vital process in maintaining blood glucose level, especially during fasting or physical exertion. So it means that in glycogenolysis, if you have a glycogen, which is the storage mode of carbohydrates, it will be break down to release a glucose molecule. And usually this glycogenolysis is take place in two major organs of the body. It takes place in the liver and in the muscles. So usually the glucose released from the glycogen in the muscles is mainly meant for the glycogen to generate energy, while the glycogen from the liver is mainly used in maintenance of the blood glucose level for other physiological processes. So now, what are the steps involved in glycogenolysis? The process involves the sequential action of enzymes such as glycogen phosphorylase and the branching enzymes to release glucose, 1-phosphate, and then free glucose from the glycogen. So you see, glycogenolysis is a sequential step that involves the breaking down of glycogen into a molecules of glucose using two key enzymes. And these two key enzymes is glycogen phosphorylase and the debranching enzymes. So what are the key enzymes? So the key enzymes in glycogenolysis is glycogen phosphorylase, which actually breaks alpha-1 for glycosidic bond, and the branching enzymes which act on alpha-1,6 glycosidic bonds to ensure complete breakdown. So see the two key major enzymes. We know that generally glycogen, if you didn't watch my video on glycogenolysis, you can go back and watch it. The glycogen is made up of the linear chain and the branch chain, and the linear chain is made by alpha-1 for glycosidic bond, while the branch chain is made up by alpha-1,6 glycosidic bond. So therefore, if you are breaking that glycogen, you must break down that alpha-1 for glycosidic bond and the branch chain. So to break to break down alpha-1 for glycosidic bond, you need an enzyme called glycogen phosphorylase. And then, of course, you need the debranching enzymes, which actually removed the alpha-1,6 glycosidic. So in glycogen, we have both alpha-1,6 and alpha-1,4 glycosidic, and you need these two enzymes to break down the complete glycogen. So these are the sequential steps. Number one, you have the glycogen. So with the action of glycogen phosphorylase and debranching enzymes, and the debranching enzyme is a bifunctional enzyme. It's a bifunctional enzyme. It is an enzyme that have two functions. Number one, to transfer some molecules of glucose from the glycogen and transfer them to the linear chain. So it has two major functions, as I said. It has the glucosidase activity. So the glucosidase activity is the enzymes that break down the alpha-1,6 glycosidic bond. That is the differential activity of the glucosidase. And then the glucan transferase. The glucan transferase, of course, is the enzymes after the cleaving, like some molecules of glucose at the branching point of the glycogen, then it will now transfer it to alpha-1 for glycosidic bond. So these are the two bifunctional activity. The hydrolytic activity by alpha-1,6 glycosidic bond at alpha-1,6 glycosidic bond, and the transferase activity by transferring the product generated by the alpha-1,6 glucosidase activity, and then it's now transferred into the linear chain of the glycogen through alpha-1,4 glycosidic. And after that, then the glycogen phosphorylase will now actually break down the glucose from the glycogen, and you now generate glucose 1-phosphates. So after you now have the glucose 1-phosphate, then the next thing is this glucose 1-phosphate can actually or can easily be converted to generate to generate glucose 6-phosphate. Of course, this happens in the muscles. So in the muscles, the glucose 1-phosphate can be converted into glucose 6-phosphate using phosphogluconeutase. And phosphogluconeutase is an enzyme that involves the change of position of a group from one position to another position. So of course, in the glucose 6-phosphate in the muscles, because in the muscles we doesn't have this glucose 6-phosphatase. So of course, this glucose 6-phosphate in the muscles will continue for the glycolysis to generate energy, specifically for the muscles. And since the free glycogen, the main focus of the phosphate group in the group is to restrict the glucose in the cell so that at least it will not be actually exit from the cell. So that is why the glucose in the muscles is usually stayed there, because there is no glucose 6-phosphatases that remove the glucose, that remove the phosphate from the glucose. That is why the glucose from the muscles never actually leave the muscle, so it's utilized there, and it's because of the absence of the glucose 6-phosphatase. And of course, in the muscles we have glucose 6-phosphatase, and this glucose 6-phosphatase is an enzyme. This glucose 6-phosphatase is an enzyme that actually remove the phosphate group. It removes the phosphate group from carbon 6. Of course, at the end, we are going to have the free glucose, and this free glucose from the liver can easily be transferred from the liver to the other cells of the body, especially blood, for the maintenance of that blood glucose level. So that is why we should understand that the glycogen from the liver is actually involved in maintaining blood glucose level. It's involved in maintaining the blood glucose level, while that of the muscles is actually involved in generating energy in the muscles, only in the muscles. And of course, this is the what you call. Oh, the next thing is let's clearly look at how this glycogen is in breaking down. So now, for example, this is a glycogen, and of course, from here, this is alpha. This is the alpha 1,4. This is the alpha 1,4 glycosidic bond, and of course, this is the alpha 1,6 glycosidic bond at the branching point. So this enzymes glycogen phosphorylase, what it does is actually is to add free inorganic phosphate. Then once you add free inorganic phosphate, then of course the glucose will be removed. So that is why here it means that from here to here, this enzymes glycogen phosphorylase was able to remove one molecule of glucose as glucose 1-phosphate. And of course, after removing this glucose 1-phosphate, it means that it will now become a limit dextrin. It means that it is now limited, so it's now reduced by 1 molecules of glucose. So then after this action, then the next one is to remove the 3 molecules, of this, like these 3 molecules of glucose that are here using deep branching enzymes. So deep branching enzymes remove the 3 molecules of glucose that are left here, and then of course, after removing it, then it will now use the alpha-1,6 glucosidase activities and cleavage. Of course, then at the end, it will now be attached at this position, or at the direction where we have alpha-1,6 glycosidic bond. So this is how it continues. Then after we have this linear chain, then of course the glycogen phosphorylase will now further remove the glucose 1-phosphate. Then of course, after that, then we have phosphogluconeutase will now change the position of the glucose 1-phosphate to glucose 6-phosphate, and of course In the liver, it will undergo glycolysis. Sorry, in the muscles, it will undergo glycolysis. While in the liver, it will actually be converted into free glucose using an enzyme called glucose phosphatase. So this is how actually glycogenolysis takes place. First, the glucose will be removed at a branching point. It's removed from the branching point, then the next thing is to remove the remnant glucose at the branching point and transfer it to alpha 1 for linear direction. So generally in glycogen, the linear change is usually have 8 to 10 glucose residues in one glycogen. So as a linear change, glucose is being removed, and then it will be moving toward the direction where there's a branching point. So when 4 molecules of glucose to 6 molecules of glucose left at the linear change, then of course the branching point glucose will start be removed. So that is where you need the action of the branching enzymes. So then the next thing after this, so this is actually the steps and the pathway of glycogen breakdown or glycogen degradation. So then the next thing is the regulation. How do we regulate this glycogen metabolism? So there is a regulatory mechanisms, and one of the major mechanisms of this glycogenolysis is tightly regulated by hormonal signals such as glycogen and epinephrine, which trigger the activation of key enzymes to initiate glycogen breakdown, ensuring glucose availability. So now we are going to look at how this glucose is being actually regulated or controlled in the body. So the major regulatory point of this glycogen metabolism. So the three major regulatory points of glycolysis and glycogenolysis involve three major mechanisms: allosteric regulation, hormonal regulation, and influence of calcium. And of course, the key major enzymes of glycogen metabolism, the key regulatory enzymes are glycogen synthase and glycogen phosphorylase. So we are going to look at how these enzymes are involved in metabolic regulation. So let's start with the hormonal regulation. So whenever the glucose level is low in the body, when blood glucose level is low in the body, then a hormone called adrenaline and glucagon as well as tyrosine, their concentration will be increased in the blood. So once their concentration is increased in the blood, then they will now go to the surface of the liver cells or the muscle cells and then bind the receptor there. And the receptor in which these three major hormones bind to its G protein-coupled receptor. So these hormones bind to the receptor, and then the message will now send into the liver cells or the muscle cells, and then the ATP will now be converted into CAMP. And a CAMP is called, or is an adenyl cyclase. And this adenyl cyclase, the adenyl cyclase convert ATP into CAMP. And this CAMP is a positive regulator of protein kinase. So what this protein kinase does is generally the dephosphorylated form of glycogen synthase 1 or glycogen synthase 1 is active and it is dephosphorylated form. So now protein kinase phosphorylates the glycogen phosphorylase 1. So once it is phosphorylated, then the enzymes will become inactive. So the glycogen synthase will become inactive. So once the glycogen synthase becomes inactive, that means that the glycogenesis, that the synthesis of the glycogen will stop. Because at that point, you only need to activate glycogenolysis because the blood glucose level is low. So you need the glucose or you need the glucose to be released so that to maintain the blood glucose level. And then of course, in the case where the glucose level is high, then of course insulin will be activated, or the concentration of insulin will be high. Then of course, once it is high, then the CA, it will now actually activate an enzyme called phosphodiesterase. And this phosphodiesterase, CAMP phosphodiesterase, cleaved CAMP into AMP. And once AMP is being produced from the CAMP, it means there is no second messenger. So if there is no second messenger, then what will happen? It means that the protein kinase 1 will not be activated. So therefore, the glycogen synthase will remain in the dephosphorylated form. So it means that it is dephosphorylated form. It means that the glycogen synthase will be active. So once the enzymes is inactive, it means that the glycogen synthesis will continue taking place. So therefore, the amount of the glucose that are present in the blood will now start reducing due to glycogen synthesis. So that is what actually happened. So in glycogen synthesis or in glycogen metabolism, insulin usually involved in making sure that the glycogen synthesis is active, so that at least more of the glucose from the blood will be transporting to the liver or muscles for glycogen biosynthesis, so that the amount of the glucose will reduce. And of course, if there is any problem, let's say there's a deficiency of glycogen synthase 1, which is the active form of the glycogen, it means that the glucose in the blood cannot be transported into either muscles or liver for glycogen synthesis. So therefore, the glucose will remain in the blood. And that will lead to a condition called diabetes. And there are so many other disease conditions, and we are going to look at them. So the major work or the major function of CAMP is to activate protein kinase. But of course, the presence of insulin activates an enzyme called CAMP phosphodiesterase, which cleave on the CAMP. So once it cleave, it will now generate AMP. And once you have AMP, it means that the protein kinase will remain inactive. Therefore, the glycogen synthase will remain active. So that glycogenesis takes place all the time. And in addition, there are also allosteric regulators. So in this point, the presence of glycogen, the presence of glucose 6-phosphates, also is a positive regulator of, or is a positive activator of glycogen phosphatase, and that is the inactive form. So once this enzyme is inactive, so it means that the presence of glucose 6-phosphate, it means that we have enough glucose 6-phosphate. So it will now actually activate these enzymes. And of course, glycogen, the presence of glycogen also is a negative inhibitor. It's a negative inhibitor of glycogen synthase 1, because it means that when you have enough glycogen, you don't need to synthesize enough of that glycogen. So that is why this glycogen will now inhibit the glycogen synthase 1. So it will now become inactive. And so also the CAMP is a positive regulator of phosphatase enzymes. Positive. So this phosphatase enzyme is an enzyme that actually catalyze dephosphorylation. It's an enzyme that catalyzes dephosphorylation. So it means that if you want to get this glycogen synthase 1 to be active, then the phosphate group attached to the glycogen will be removed by synthase phosphatase. So the presence of insulin is a positive regulator of synthase 1 at the same time. So after cleaving CAMP to AMP, it also activates phosphatase by removing phosphate group to make sure that the synthase enzymes is active. So this is actually the allosteric as well as the hormonal regulation of the hormones, of glycogen metabolism. So therefore, the phosphatase enzymes, or the synthase phosphatase here promote glycogenesis. while the kinase enzymes here promote glycogenolysis. So then the next thing, what is the effect of calcium? So calcium ions or cyclic AMP act as a secondary messenger. They act as a secondary messenger, and these are examples of negative control. So the calcium ions usually activate phosphorylase kinase. And of course, this kinase enzymes is an enzyme that activate glycogen phosphorylase. So once glycogen phosphorylase is activated, it means that, of course, the glycogenolysis will be activated, and it inhibits CAM, it inhibits glycogen synthesis. It means that it will inhibit glycogen synthesis. So calcium ion is actually a secondary messenger that promote glycogenolysis and inhibit glycogenesis by inhibiting glycogen synthesis and activating glycogen phosphorylase, which is the master enzymes of glycogenolysis. So of course, this is the actually allosteric regulation is a similar mechanism to glucose 6-phosphate activate glycogen synthesis, and of course, glucose 6-phosphate and AMP inhibit glycogen phosphorylase, and calcium ion AMP also activate glycogen phosphorylase. So it means that calcium and AMP they promote glycogenolysis. Glucose 6-phosphate and ATP also inhibit glycogen phosphorylase. So therefore, it inhibits glycogenolysis. While glucose 6-phosphate activate glycogen synthesis. So this is actually in the process. And of course, in the liver, the glucose 6-phosphate and ATP are they actually negative regulators of glucose 6-phosphatase. And of course, glucose 6-phosphate is also a regulator of glycogen synthesis. So this is actually the allosteric regulators in the liver, that of the muscles. So of course, the next thing is the clinical relevance of the glycogen. So glycogen storage disease generally are inherited metabolic disorders affecting the glycogen metabolism, leading to abnormal glycogenolysis process. So we are going to look at them. There are so many of them. So we are going to look at them in my next video. And we have hypoglycemia. So excessive glycogen breaking down can result in low glucose level, causing symptoms like dizziness and confusion. So hypoglycemia is actually associated with because diabetes. Altered glycogenolysis contributes to glucose dysregulation in diabetes, impacting blood glucose control. So we are going to look at them in my next video in detail. So what are the impact of energy metabolism? So 40% of muscles glycogen utilization, so approximately 40% of energy needs are made through glucose production via glycogen or via glycogenolysis, which is a crucial during fasting or intense physical exercise in the muscles. That is muscles glycogen utilization. So about 40% of the energy needs in the muscles depend on the glucose generated from glycogenolysis, which usually occurs during fasting or extensive physical exercise. And glucose production, around 30% of glycogenolysis contributes to energy demand during muscle contraction, supporting exercise performance. And of course, in the liver glycogen utilization, the liver glycogen breakdown accounts for 30% glucose supply, maintaining blood glucose level for various physiological functions. So that is why we say glycogen metabolism in the liver or glycogenolysis in the liver is meant for maintaining blood glucose level. And that of the muscles is actually for energy generation, specifically for the muscles. And then, of course, so glycogenolysis during glycogenolysis in exercise. So enhancing performance, efficient glycogen breakdown, and glucose release enable optimal muscles function. So delaying fatigue and enhancing endurance during workout. So if there is an efficient glycogen breakdown, it means that there will be an efficient release of glucose, which make the muscles function perfectly without any delay. And this leads to enhancing endurance during the workout. And during exercise, glycogenolysis in muscles provide a rapid source of glucose for energy production, sustaining through long physical performance. And of course, these are the summary and the conclusion. So glycogenolysis is a crucial metabolic pathway that regulates glucose homeostasis by breaking down stored glycogen into glucose. So understanding its process and regulation is essential for metabolic health and energy metabolism. So, ladies and gentlemen, this is where we are going to stop, and we are going to meet in the next video where we are going to talk about glycogen storage diseases. And thank you for your time. And if this is the first time you are coming to my YouTube channel, please subscribe.